Hello everyone, my name is Sumit Suresh Achalya. Our today's topic is DNA, the discovery of DNA. It is amazing how two important things are being discovered by person who spend most of their lives dedicated to research. It is perhaps more amazing how two scientists discover one thing while trying to discover other things. Imagine if no one was patient without the conduct experimental to prove their assumptions. If that was the case, we would not know today that DNA is the genetic material. This paper will discuss how DNA was discovered, its structure, and the scientists who contributed to the discovery. Deoxyribonucleic acids DNA. DNA is considered as the molecule blueprint of life that can be passed to the next generation. It was discovered in 1953, University of Goregaon, 207, from experimental seeking to understand bacterial phenomena which claimed many lives during the 20th century. During this experiment, research submitted into discoveries, which later on become the reagents for further experiment, leading to the discovery of DNA as the genetic material. 0 Canara 2008. DNA looks like a twisted loader where runs are separated by 2 out of 4 molecules that are interlocking. These molecules are nucleic acid bases. The 4 molecules include thymine, adenine, cytosine, and guanine. University of Goregaon 2007. Certain scientists have been notable for conducting experiments leading to the discovery of DNA. One of these was Oswald Aware, whose team was trying to find out how to treat laboral pneumonia. For his experiment, he proved that DNA indeed carries genetic information. During that time, many people believed that the gene would be made up of protein and not nucleic acid. Another scientist which contributed to DNA discovery was Frederick Griffith, who in 1928 conducted an experiment utilizing pneumonia bacterial and mice. Recent studies point that Griffith experiment was one of many other hunting that DNA was the heterodytic material. Gibbs estimated from his experiment whenever he used a smooth and wrought strain of Streptococcus pneumonia on the mice that polysaccharide chloride coating was the case of the lineless of the mice. Further into his experiment, he found out that some things in the living cells and not the polysaccharides caused the disease. Later on, he separated that there are certain bacteria he injected into the mice has absorbed the dead yes strain bacterial genetic material. Furthermore, he speculated that the protein contains in the chromosomes was not the genetic bacterial due to finding that heart denaturated proteins biological or claminant college. At first, Aware Very was skeptical of the result of gene experiment. However, other research further studies Gibson's findings. In 1931, Sy and Dawson found out that transformations, a process wherein one strain absorbs the genetic material of another strain and becomes that bacterium. This is about DNA structure. Thank you.